want to welcome everybody to this ongoing series in cell and gene therapy. This is our, I believe, third year. Am I right, Katie? Is that right? I think we've been doing this 3 years now.
I believe 3 years with different focuses and discussions. But yes, 3 years.
Excellent. Yeah. Well, I'm glad at least I got that straight for today. So I'm going to introduce, as Katie pointed out, every year we try to get a new group here. And what we'll be doing, and just so everybody remembers, this is the actually recorded / virtual session of Interphex Live, which usually comes to you from the Crystal Palace at the Javits Center. And obviously, as you can see, looking around, we're at Javits Center. But the show will be October 19th through the 21st, and we look forward to meeting everybody there. So our advancements in cell and gene therapy, our speakers today are Audrey Greenberg. Welcome, Audrey. She's from Discovery Labs. And then we have Dana Hamill from Descartes, Graham Tulloch from J&J, Norm Goldschmidt from Genesis AEC, and my friend Katie Worrilow from LifeAire. Just as far as background goes, Dana is actually the founder of Descartes and as the chief operating officer. And prior to founding and establishing Descartes, she served as the director of strategy and business development for the Center of Cellular Immunotherapies at the University of Penn, Perelman's School of Medicine. And just for the record, during that time, Ms. Hamill managed the Penn-Novartis Alliance, and that total item is going to go down in history. It's in my history. It's in my background. Thank you there. Katie Worrilow. Katie and I have known each other a few years, and Katie's background is essentially she is the founder and CEO of LifeAire. Now, you would ask, so what's the background? So let me give you the story and the run up to that. That she has her doctorates in anatomy and cell physiology from University of Virginia School of Medicine. And then she completed her postdoc at UPenn School of Medicine. You start to see the commonalities here among our members. And there she served as scientific director of the in vitro fertilization program for over 20 years. And she did an extensive amount of work in the interactions of and impact of ambient air on the successful human embryogenesis. And that work, along with her colleagues, has led to her establishing the unique technology and the company that we have come to know as LifeAire. And in addition, this time we have Audrey Greenberg, and Audrey is the co-founder of Discovery Labs. And obviously, when the other commonality is she happens to be sitting right in the Philadelphia Silicon Valley. Go PA. We kind of PA centric here. So having said that, just want to give some background. And so Audrey is was the co-founder there, and that's the Center for Breakthrough Medicine. It is a 2.4 million square foot integrated scientific hub. And as you can see the background there, Audrey's giving us a little view there. And CBM is a contract development manufacturing organization, or as we prefer to call it, a CDMO, and that enables rapid development and commercialization. And finally, we have, we still got Graham. Graham is scientific leader at large molecule development and biotherapeutics department at biotherapeutics organization at Janssen Pharmaceutical, which is a division of Johnson & Johnson. Graham has a strategic responsibility for driving and maintaining scientific capability through biotherapeutics development with a focus on biologics, drug substance, including therapeutic proteins and cell and gene therapy. And Norm. So Norm has over 35 years of experience spanning all types of facilities and processes necessary to bring life sciences to the commercial forefront. In that, those range from R&D facilities right down to mass production and commercialization. He is president of Genesis AEC, which is an engineering and consulting firm, and they also do quality CQV special in that area. So having said all of this, as you can see, we got a powerhouse going here in cell and gene. So, every year we try to put a little frame around where everything's going. So, Audrey and I talk a little bit. And so I think, why don't you start off, give us a little background and see where we go from there.
I think in cell and gene, we're really at the beginning of a medical revolution being driven by the ability to cure rather than treat disease. And as we all know, there are many complexities in this path to commercialization for advanced therapies. We're here to discuss those complexities and the key considerations to take into account in the development and manufacture. So to kick off the discussion, we'll do a quick round robin with each of my distinguished panelists relaying what they see as challenges and complexities in this process. From my perspective, I started the Center for Breakthrough Medicines just a couple of years ago to really address the severe market-wide capacity constraints in development and manufacturing, as well as some of these extreme challenges, which from my perspective include, first, current therapeutic platforms are developed in academic labs, really challenged by the ability to efficiently scale and reproduce and manufacture these therapies. Second, the science behind these therapies is constantly evolving and changing, and the regulatory landscape really needs to keep pace with these changes. And then third, while there is there's investment in the therapies, but there's really little investment in platform technologies and industrialized manufacturing solutions. This results in headaches and really bottlenecks in the path to commercialization, including speed bumps in tech transfer and process development, and really just inefficient scale-up models, which results in headaches down the road. Thanks. I'll hand it over to Norm.
Thanks very much. You hit on a couple of the same items that I'm seeing. It is this transfer from institutions of higher education and healthcare without a real CGMP bent, where the barriers to commercialization can be huge. Along with that, so we're working to create facilities and processes that scale off of the lab bench and into a potential clinical and commercial application. Along with that, we have the developing regulatory environment, which very, very strongly led to a lot of our European agencies have really taken a strong stand on cell and gene therapies, which really are such a broad number of different technologies. When we say ATMPs or advanced therapeutic medicinal products, we're talking about cell and gene therapies, cell therapies that can't be sterile filtered, gene therapies, which maybe are vectors that can, oligonucleotides. There's a whole series of different products that we've lumped together. And unfortunately, I think the regulatory environment is evolving behind the technology. The great news from my perspective, designing these facilities and processes, is that the technology is moving so fast. We are seeing great process closure. We're seeing advancement in even the small scale autologous cell therapies, where we're getting out of the sort of lab bench, everything's open to the environment, and reducing the impact of the background. And that really feeds into some of the discussion that Katie and I have had for several years now about the importance of understanding the background environment and its impact on the product. Hopefully, as these technologies mature, we're going to see less and less impact of the background, but it's still really important to understand. I guess, Katie, I'll kick it off to you.
Okay. Well, very, very well said, and I couldn't agree more. As Russ shared, I'm a trained reproductive physiologist, and I've really had the honor of serving as a clinical provider of human in vitro fertilization, or the culture of the living human cell outside the body, for up to 6 days. So I'm extremely cell-centric, if you will, and have really spent the last 25 years examining the molecular epidemiology or molecular role of the culture environment, as Norm just described, towards the successful in vitro culture of the living cell, including not only the ambient air that surrounds all of the processes in cell and gene therapy, but also the medical gases that are serving the incubators, the bioreactors, open versus closed environments, and the impact of very specific airborne chemicals, your volatile organic compounds or VOCs, as well as your biological, your viruses, bacteria, fungi, the impact of these specifically on successful cell growth, cell expansion, cell potential, cell viability, and ultimately the successful delivery of a cell solution to the patient, which is the ultimate goal. So I'm going to quote one of our fellow panelists, Dana Hamill, who so eloquently shared just recently, manufacturing cells is an art. Manufacturing cells is an art beyond SOPs. I could not agree more. And therefore, what I see as a new and fairly unrecognized challenge, and knowing the impact of the environment, the proven impact of the environment on successful cell culture, the challenge that I see is to provide the airborne metrics deemed optimal despite the ongoing activities in that space, your SOPs, your cleaning protocols, how you bring the new materials into the space. So a balance needs to and can be achieved between your ongoing cell processing activities, maintaining the proper environmental metrics, but yet balancing those with the needs of the living cells. So this balance is achievable, and it has been proven to significantly increase successful cell metrics and delivery to the patient. So this is the challenge that I see, but fortunately, we have a lot of data, we have a lot of direction, and there are answers. So Dana, since I quoted you, I guess I'll pass the baton to you.
Thank you for that. And I maybe I should pay a little more attention to what I say sometimes. But thank you, thank you for that. And I think a big reason that I make that statement is that many moons ago, over 20 years ago, when I first started working in manufacturing, I was making clinical grade adenoviral vectors for clinical trials at Penn. And I remember how important it was that I actually went in to look at the cells to see if they were ready for harvest. It wasn't a timing, I knew it was within a particular range that I needed to harvest, but I really needed to see what was happening in that Petri dish to be able to make a decision about whether harvesting was optimal so I could get the best yield. And so doing that at a smaller scale in an academic institution and kind of moving that into a commercial scale, I think remains a challenge for everyone in the field. And so as I was listening to you, Katie, and Audrey and Norman, I was thinking about, some of the other challenges that are out there. And, I definitely think that kind of tying into manufacturing being an art, we have a human capital shortage. And so we have talented scientists that are in the area, and yet they need to be trained in GMPs. There's a code of federal regulations that they need to understand. They need to follow SOPs. They can't be using a pencil and kind of being, flip about it. These things are really important. And so, and then there's this turnover of that talent, right? So there's a competition for it as well. So you have to attract and retain them, and then they're in Silicon Valley, and they have opportunities before them, and it becomes a really big challenge. I know it was a huge challenge at University of Pennsylvania. We would bring folks in, we would train them, and then they get a really nice financial offer from another startup in the area, and those are salaries that a university doesn't really compete with. And so it became actually a major issue in the area. And so I still see that as something we need to face. I also see manufacturing facility availability as a continued challenge for us. And what I mean by that is there are more CMOs and CDMOs cropping up in the area, which is fantastic for the field. However, there's still not enough, because we need to make GMP plasmids, we need to make GMP viral vectors, we need to make GMP cells. And they're all a little bit of a specialty. They don't all happen under the same roof. And the wait lists can be out over a year. So you can't even launch your clinical trials because there's no availability to manufacture. As was already stated, the science moves so rapidly. By the time a year goes by and you get your slot in to manufacture vector, the science could have changed. And I think another key challenge in the area that many face is just the cost of goods, right? So there's this whole allogenic approach versus autologous approach. And many in the field feel like auto is out. We're all going to move towards allo. We're going to put all our investment dollars in allo because that makes the most sense. However, clinically speaking, I think the jury's out, and I think they're both very important. But we do need to address what that impact is going to be for the healthcare economics for treating patients with these therapies.
Graham, I think, do you want to weigh in, please?
Yes, and maybe to echo some of what we've heard, and the recent successes in cell and gene therapy has created a tremendous hope. Hope to provide effective treatments for diseases that were previously incurable. But that hope is hampered by what I see as significant CMC challenges, and those challenges need to be overcome to enable broad patient access. So one of the areas that are building on what Dana said in terms of the success has been autologous cell therapy. I mean, that's tremendous. But the processes are complex, they're costly, difficult to manufacture. One patient, one batch approach does not provide opportunity to leverage economies of scale that we've used elsewhere. So solutions to that need to be found. And then sort of building on the comment of autologous, allogeneic. So allogeneic on paper looks good. I think Dana's points about the clinical jury is still out are valid. I think the CMC jury is still out on that as well. And it can move from one patient per batch to maybe hundreds, perhaps 1,000 or more patients per batch. But the solutions are not easy to manufacture. So those will need to be overcome as well. In the case of both gene and cell therapy, viral vectors are often used. Those themselves are difficult to culture, difficult to manufacture, difficult to scale up. They can rely on simultaneous transfection with multiple plasmids. Purification can be challenging. Processes, both from a viral vector perspective and a cell therapy perspective, rely on manual processes, labor intensive, requiring specialized skills that are in short supply. All of the things I think that Dana had mentioned. And together, these present a significant challenge that the industry needs to overcome to realize the full promise of cell and gene therapy.
I agree. And we're an industry that has a product in general. We have a product that's waiting for a patient. In cell and gene, it's a patient waiting for a product. So, I don't think it's hard to get it through people that we're trying to progress to that point. All right. As we move along, but again, these are things that are, slowly coming around. So Audrey and Dana, I heard a couple of things. I want to go back and let's talk about them. And, we've heard a lot about all these constraints and limiting the increase in cell and gene manufacturing capacity. Heck, we've heard that before cell and gene. If you recall, I would say not a lot of years ago, it was like, biologics manufacturing had a cap on it because there was not enough bricks and mortar out there. And then we got by that with single use and all the rest of it. But in your opinion, what are the three most scalable obstacles that will yield the most capacity within the near future? So Audrey, why don't you start off?
Certainly things that are preventing growth are what Dana mentioned. The first thing is just having the actual space to manufacture in. And then the other thing that Dana mentioned was having the talent and the expertise to be able to do the operations that are required to manufacture these therapies. And then third, I think it's something that Graham touched on, which is having the technology to do a repeatable, scalable process, including perhaps automation to eliminate some of the human element that's involved in the manufacturing process. So I think those are the three things. But I'd be remiss if I did not mention the fact that there's an incredible amount of capital required to build these facilities. And if they were not so expensive to build, we would probably have many of them. But we're talking about, somewhere between $400 to $2,000 per square foot, depending on the space requirements and the equipment that's involved in that space to build these facilities. So to build them in a speculative way, to sort of get in advance of the need that's required is important, but it requires those that are willing to take the risk. And I think given some of the market dynamics that we've seen recently, that can be addressed. And so that's really what we're doing. The Center for Breakthrough Medicines is building a tremendous amount of GMP manufacturing capacity, as well as expertise and equipment in plasmid DNA, viral vector production, and cell processing, including the analytics, inline analytics, and product release analytics that are required to really get through the process and reduce the supply chain constraints that we're seeing that were exacerbated in the pandemic and in the current market environment.
Yeah, Audrey, I completely agree. And as you were talking, I was thinking of, some others, but I think that you really nailed the main obstacles. I know automation would be a huge help to the field. There's so much human labor involved that if there's a way to relieve their work on some of the more mundane things, or what appear to be mundane, and have them more focused on things that require their talent, in their brain, then that would be really useful. And some of it is very repetitive. And so, seeking automated solutions, I think would be a huge help to the field. But with automation comes cost, right? So you touched upon the cost thing, and I know that further on down the line, as we think about how much these therapies cost, part of the reason they're so expensive is because of all of the infrastructure that has to be put into place. The talent that you hire are people that have usually multiple degrees, and so they're more expensive to bring in but are needed. And so the whole thing is costly and is worth it, I think, to our loved ones, to our public. We are so close to finding other ways to conquer cancer. Like, let's put it all in there. But it's a expensive and challenging endeavor. And you touched on the talent thing again. I can't emphasize that enough. I sat on an advisory board for the Commonwealth of Philadelphia, where Philadelphia as a city had come together and talked about, how do we address this for our region? Can we create new programs in the community colleges? Can we create new programs with better certifications? Like, how would manufacturing folks respond to that? If you had somebody coming out of school with this particular type of certification, would you hire them? And then we even talked about the bricks and mortar piece. Like, do we need more manufacturing facilities? Should Philadelphia have a manufacturing facility that's available to the people in the community, even those who are startups and just need something smaller? And then there's all the intricacies, as many of us know, with manufacturing facilities themselves, right? So it's not a one size fits all, right? You don't make virus in the same room that you're making CAR T cells, so you don't even make them in the same building. And then there's, the air pressure and the airflows and the particle counts, and there's all of these different things that go into, manufacturing a virus versus manufacturing a cell or a dendritic cell vaccine or a CAR T cell. I mean, there's all different kinds of applications of cellular immunotherapies. And so thinking about these CDMOs, is it a one-stop fits all? And it's just this massive facility that's a big campus, or do you specialize and have them in different places? And I think some of these things are still getting worked out. And thank goodness for that, because we certainly need it.
Let's just, I see where, the folks in Silicon Valley that are trying to get their arms around the most important piece, and that is the talent. So how do you manage the turnover piece? I mean, that's insane. I mean, by the time you get somebody ramped up to where they're really providing value to the system, they walk away. I mean, so what do you do here? Do you incentivize them? Talk to us. What have you been doing?
Yeah, so I can speak to it more from when I was at University of Pennsylvania, and we had a GMP facility there. It's still there. And we would hire folks to come in and work in the GMP facility, and we would train them. A lot of times these are kids coming out of the local universities, maybe someone who came from, a university further away, but these are people who didn't typically have a lot of GMP experience, or maybe they just had some cell biology experience. So the training program for these folks was at least 6 months of intensive training, which obviously requires time and effort from other staff to train these folks, to get them proficient in the skill. And then sometimes what ends up happening is, one of the local startups, or maybe a more established biotech company in the area, it could be down in the Navy Yard, it could be over in University City, but they're hiring. And you know what? They pay a lot more money. And the university has a kind of package program that they offer to employees, tuition benefits, outstanding 401k, outstanding health benefits. And these smaller biotechs don't have all of that overhead, so they can give more cash in the paycheck, right? And some stock options. And this sounds really sexy, especially if you're at that place in your life where you're still able to move around, you don't have children yet. And then all of a sudden, we're like this training boot camp for people in the area to start. So they're always leaving, but we're not actually bringing them in, because to come in would be a pay cut. So we actually put together a committee and really looked at bringing in some advisors to help us figure out how to change the hiring dynamic for those particular type of roles. But it was really it really helped us see just how competitive the space is, and how much time and effort goes into training. We associated costs with all of that, and how much money we would lose every year by training these folks and then having them jump around. So I think it's definitely a challenge in terms of the employer, in terms of trying to run that business. And some of them unfortunately hop around, and they're just kind of accumulating the information around the SOPs and then taking that someplace else. And so a lot of times it's the know-how that's really important in this field. So you can't take the SOP, but you can take the know-how with you, and that's a little bit harder to control. So I think it's a real issue for all of us.
Yeah.
And something we should all kind of come together and work hard at making sure that, if somebody from Audrey's group is coming over to my group, I need to call Audrey and we need to talk about that.
Yeah, I think that, and and again, there's more to retention than simply, payment and 401ks. I mean, there's got to be a policy about it,
If I may, Russ, it's about the mission, right? It's about being part of something that you really believe in. And it is both the strength and weakness of being in a great ecosystem like this, right? Where there is the talent, where because you've got the universities feeding people in, and you've got all these attractors to the talent that it takes to do this kind of work, that does come with the downside. I mean, if you want to lock the people in, put them in a remote location where there is no attraction, but then how do you get the best people to do that? So I think there is a natural sort of, I think of it, I'm sorry, I'm an engineer. I think of it like vapor pressure, that you're going to have people getting liberated all the time and folks coming back in. But if in fact you have a compelling vision and a compelling mission, I think that locks people in better than most anything else. And I'm sure other folks here can attest to that.
Yeah, you want to have a culture of respect for the individual where they have a career track, especially with millennials. They like to see how they're going to advance. They're very focused on title, work from home, other benefits. So offering a compensation package that includes consideration for individual circumstances, especially coming out of the pandemic where working women were really responsible for childcare at the same time. So having the understanding and the flexibility to work with your workforce, having policies and procedures that can address some of those needs, I think makes a big difference too.
Well, I think so. But, I mean, speaking of environments, all right, I mean, like we always say on this group, we can spend on one topic the entire hour. But what I wanted to touch on, though, was Katie, getting back to, we're talking about expansion. Let's look at it. We're talking about environment, but that's what triggered some thoughts in my head. And that is, if anything, the environment in which we're trying to manufacture, especially within heavy city limits, Philadelphia, San Francisco, Los Angeles, where I'm at is, the environment ain't getting any better outside. So, from your experience, I mean, we're talking about a lot of, living cell cultures and all this. And where do you see cell and gene and CAR T and all of this? How do we maintain and protect the environment in which we're manufacturing going forward now?
Well, there are a number of things that can be done. And, when Norm just mentioned, working towards a common mission and vision, whether you're culturing the human embryo or the patient cells in cell and gene therapy, these cells carry an exceptionally high therapeutic value. And in the lab, we have one chance. We have one opportunity to successfully grow these cells for hopeful delivery and successful delivery to the patient. So some of our learnings in the culture of the human embryo, some of the pearls that we've learned through negative data, and I personally never think negative data is negative. It always teaches you the next step of improvement. But what we've learned the hard way is that you have to maintain. And Russ, you just talked about outside air and the dynamic nature of that and how that's probably, unfortunately, not improving. But there's also the inside air, just by virtue of what we're doing in that laboratory, those ongoing SOPs. So what we've learned the hard way is that those exceptionally low levels, parts per billion levels of cytotoxic VOCs, and really below detection viables, your bacteria, viral, and fungal pathogens, must be maintained on a consistent basis. Not just when people have exited the building and the SOPs have calmed down, whether it be in a night shift, but they have to be maintained continuously. And we've been asked to perform fairly extensive environmental testing in cell and gene therapy manufacturing spaces, and we found extraordinarily high levels of some of these more toxic VOCs within the manufacturing spaces, whether it be the purification area or cell expansion, kind of throughout the different spaces. We learned which VOCs were most common to cell and gene therapy, often the sources where they were coming from, and then from our own research, studying the impact of specific VOCs on successful cell culture, we've learned that even very low level airborne VOC concentrations, they actually partition, and we're actually finishing a manuscript on that right now, but they actually partition from the gaseous phase. So let's say you have 100 parts per billion of IPA, isopropyl alcohol. They can actually partition from the gas phase into the liquid phase, or your cell media, and then within the cellular phase. And depending upon the VOC, and there are over 90,000, depending upon the VOC, they can make that travel path within fractions of seconds. So how does that happen? Each VOC has its own unique biochemical fingerprint. It can be polar, apolar, high molecular weight, low molecular weight, biogenic, as well as each carrying a unique, what's called a partition coefficient. So these VOC specific factors are going to dictate how quickly that VOC goes from your air, the air surrounding your procedures, from the gaseous mixture to the liquid phase, your cell culture media, into the cell. And once present in the cell culture media, and obviously within the cell, there is no diluting out that can be done. It's a permanent threat to that cell. And there are multiple highly respected publications demonstrating the cellular impact specific to certain VOCs, impaired mitochondrial function, increased intracellular calcium, decreased spindle formation, increased oxidative damage, all of this which is impacting cell viability, cell growth, and ultimately cell potential. So given the high levels of cytotoxic in-space VOCs that have been identified in some of these manufacturing spaces, in particular, now this is just a short list, but in particular acetonitrile, formaldehyde, IPA, ethanol, given the list of cytotoxic VOCs and the high concentrations, and the fact that we know VOCs cross a phase transfer from gas to liquid to cell, and we know how quickly each of these VOCs accomplish that, and we have the proven impact of VOC toxicity, it's really critical from our learnings and our experience on the cellular and the clinical side. It's critical to drive this variable to zero during operations within cell manufacturing.
It's interesting, Katie. So let me just ask Norm. So when you build these facilities, Norm, are your clients and the contractors and the folks you work with, are they taking all of this into account, or how are you doing facility design to account for this?
Well, regrettably, I think quite often it's not been accounted for. And I'll take a step back even just to talk about some of the larger, well understood contamination sources. When we talk about viable and non-viable particulate matter, as well as the molecular contaminants, often what we see is facilities are designed with those sort of old rules of thumb, some air change rate, a bunch of HEPA filters, and we call it good, without accounting for airflow patterns, sources for particulate matter and for viable matter within a space, the gowning, the cleaning protocols, and the like. So from my perspective, it really demands that we take a step back and we understand the process, understand, and this very much, Katie, what you were alluding to, if the cells are really our client here, if the cells are what we're trying to protect, then we have to think about the process and the particular susceptibilities of the cells, of the process, and start designing the products, start designing the facilities around the product and around the processes. So it means understanding the sources for contaminants, whether they be particulate, viable, or molecular. And then understanding how we abate, how we control, how we manage. And that includes the gowning, which is really, really important to control of viables, and the cleaning, disinfection, and then their impact on the environment around us. The thing that Katie didn't say was we put those VOCs into the space to do some of these tasks like disinfection. So it's important to understand how they get there, how much, and how we get them back out again. And that really reflects back to what you were talking about, Katie and her team, where they're focused very much on each one of these things, the viable, the particulate, and the molecular contaminants. Just as a quick aside, I think most engineers who design clean room facilities don't even realize that there is ISO has a standard for molecular contaminants within the 14644 series of clean room standards. So it's just something that I think folks have, as I said, are commonly just throwing some rules of thumb at it. And there is opportunity. There's great science and great engineering available to work through, understand the challenges, and debate them. And that's where, quite frankly, some of this equipment, specialized equipment, comes into play. And that's really what Katie does.
Mm-hmm. I tell you, one of the things that Norm, you bring up, and Katie, I mean, one of the things when we plan tech transfer, especially on my end, where we did mostly small molecule, more let's call it biologics in the large molecule, stuff that traditionally. One of the things I always tell the folks and the guys that work on a team is, it's not so much the transfer as in the place I'm going to land. Where am I going? What do they got? There are so many dimensions to it. And I'm not even going to get into that. But Audrey, why don't we talk about that for a bit? I mean, to me, the points that Norm made and Katie made, that's part of that because that's where I'm going. But there's a heck of a lot more there. So let's talk about, if I got something, I'm going to transfer it out. What are the challenges?
Yeah, I mean, I think clearly, as we talked about at the top of the call, the needs of cell and gene therapy companies are much more technically challenging. And because a lot of the therapies were developed in labs without consideration necessarily most of the time for CMC, the process development aspect of the tech transfer is much, much more intense. So someone can bring to you, let's say a big pharma has a repeatable process that's well vetted. They can tech transfer in. We at the Center for Breakthrough Medicines, we really have an open door partner and plant philosophy where we will work with you very closely, tech transfer your process in, and then it's off to the races. But for some of these smaller companies that haven't done scale-up manufacturing, the tech transfer process is much more intensive, right? We're visiting, if you have a plant and you have a process now, we're coming to visit, observe, get educated, tech transfer in that process, work with the therapeutic company to make sure that we're all on the same page, that we're working together towards the same goal, and we're meeting the timelines that are involved, but also creating not just a process, but analytical methods that can prove comparability, potency, and some of the key items that the FDA is very concerned about. You have to have these analytical methods developed early. We're seeing a lot of dings right now in the commercialization and approval process because of the analytical methods. So I think it's process, analytical methods, and certainly having a team of people that can execute on that is going to be really important with a culture that's open to collaboration and partnership.
In the environment of tech transfer, I mean, there's a lot going on. I mean, Graham, so let's say, in your situation and, and your responsibilities at Janssen/J&J, I mean, so what do you do? I mean, are you going to change it? You're going to go through, what goes through your mind here? I mean, how do you implement, like raw material? I mean, there's so many facets here, Graham. So at least take a shot at it.
Oh, yeah, thanks, Russ. Changes in biologics manufacturing processes are common during clinical development, but they also come with risk. For the most part, the changes are focused on making those processes commercially viable, ensuring that the drug is available with the quality and at the quantities needed to address patient needs. I always find it helpful to weigh the risk of making a change against the risk of not making a change. And broadly speaking, there's two large categories of risk that consider doing this, a technical risk and a regulatory risk. So technical risk focuses on the ability to make that post-change material consistently, meeting quality, cost requirements at meaningful scale and throughput, all the things that Audrey had described. And there's lots. There's the tech transfer. There's introducing equipment to reduce manual operations, closing the process, changing manufacturing sites, changing process designs, changing raw materials. So all of that has to be looked at. On the regulatory side, what we're doing is balancing the challenge of being able to demonstrate comparability pre and post change against regulatory requirements. And key to the comparability perspective is being able to analyze what the impact of the changes are. And I think everybody in the audience knows that cell and gene therapies are inherently complex. So development and validation of analytical methods to demonstrate comparability can be challenging. Understanding the potential of a change and what that impact could be on quality, and then selecting appropriate methods. So that all has to be factored in to managing the change. And then on raw materials, right? So there's a number of relatively recent guidance that different health authorities have put out on raw materials, and they're not harmonized. They will categorize raw materials differently. A plasmid can be categorized as a raw material, but it can also be categorized as a critical starting material. Viral vectors as drug substance or starting materials. And this leads to different reporting categories, different levels of evidence to support the change, different descriptions and control strategies and regulatory filings. So all of that becomes quite complex and has to be factored in. To minimize this, it's desirable to get change done, notable changes done prior to entering pivotal clinical manufacturing, and then manage the technical development programs so as to provide information necessary to ultimately receive approval from health authorities. And for this, we find that close coordination between CMNC, between the clinical and regulatory affairs, helps to assure that balance of risk with supply, and then ultimately, communication with health authorities to help shepherd through those changes all help.
One of the things that I think that's important, that we still have to, there's so many fine differences, raw material versus critical starting material, dissimilarities in the way people understand things. Again, so Katie, let me get back to the environment. And I think this is something that, if I'm in a clean room and I'm making it in a clean room space, let me see if I got it straight. There's still, the environment still poses a threat to success in a living cell environment. Did I get that right? I mean, that's my question.
Yes, absolutely. From our experience, I mean, we performed human IVF in an ISO5, a certified, quarterly certified ISO5 clean room. That was, it was designed to be built as the perfect space for cell culture. And we had adequate air changes per hour, positive pressure, remediation of viable and non-viable, successful quarterly certification. Again, it was and we also had 450 pounds of carbon and potassium permanganate, which is a lot of molecular media to try to remediate VOCs. But as we learned, and again, you don't know what you don't know, but as we learned, carbon is going to remediate a certain biochemical family of VOCs. Potassium permanganate is going to oxidize another biochemical family of VOCs. But there are thousands of other cytotoxic VOCs that were able to bypass the 450 pounds of media that we had in place and enter our cell culture laboratory. Granted, at parts per billion, but that's all that's necessary to be toxic to the living cell. So again, our learnings were, it's a very tight range of low-level PPB VOCs that have to be maintained during operation, which can be challenging, but is doable. The other thing that we learned was about the outside air and the percentage of outside air or supply air that was feeding our laboratory, which is very typical. It can range from 20% outside air to 100% outside air. And obviously, Norm would be the best to speak on this. But we learned the significance or how dynamic the outside air is, just given common equities. And in some of the cell and gene therapy environmental testing that we were asked to do, they would tell us that, we see significant drops in our cell metrics after a Tuesday or after a Thursday. They would tell us these patterns that they would see, which we had also experienced. And what we learned through the extensive testing was that on Tuesdays, that happened to be the day they would exercise their generator. So given the location of their generator and the location of their outside air intake, they were seeing the ramifications of the VOCs being generated from that exhaust into their cell culture space. Other days, patterns that they saw, one that I remember coincided with a fairly busy day of truck deliveries for their supplies. And so these truck deliveries, often the trucks would idle, just stay idle, and the engines would continue to run. The exhaust, or some of the exhaust, then entering their intake and then having the impact downstream on their cell viability. So the percent outside air is key, but whether it's 20% or 100%, it needs to be controlled. You don't want, and I speak to this from experience, you don't want an outside activity cutting the lawn of a soccer field or resurfacing a road or initiating construction. Those in the lab, those in manufacturing will never be able to control the activities that are happening outside. So what's key in my mind is to protect your cell processing events, protect your success from anything happening both outside and within your own staff. The last thing, well, there are many pearls from this clean room that we learned, but it all matters. So even the fabric of the laboratory, again, which, there's no one better to speak to this than Norm, but your walls, your ceilings, flooring, gasketing, the equipment, steps to repair the equipment, steps that we would take to repair our equipment, even adding small gaskets, those can off-gas cytotoxic VOCs for years. New equipment, we learn to order, if you can do this, order your new equipment in advance and let it off-gas outside of your laboratory. Anything to reduce the VOC burden and the contributions to your lab space. And the last thing, I did say that was the last, but I thought of one other thing that was significant to us. The medical gases that we chose, we paid a premium for the highest grade medical gases available to serve our incubators. And what we learned was, despite the premium paid on these medical gases, the lot sheets would show significant levels of cytotoxic VOCs. And so these were feeding our incubators. So as hard as we were trying to control the ambient air, the outside air, our own SOPs, and provide a balance with the needs of the living cell, we also learned that the medical gases were contributing to that VOC load, during our cell culture period in the incubators. And so that was another significant learning. We also learned that as the tank would start to dip below 300 PSI, those VOCs are concentrated. So we would often, despite the ramifications financially, we would often stop using a tank when it hit 300 PSI, just to avoid those concentrated VOCs.
Well, I got to tell you, as we go through this stuff, I mean, you guys always give me more headaches than I expected. So anyway, I want to bring this around, and I think what we need to do is, I think the first thing we got to do is, Audrey, and so I'm shopping for a CDMO based on all those horror stories we've heard. I mean, what's the insight here? Obviously, it's all going to wind up on your back. So why don't you give a little insight in that area?
Yeah, definitely. I mean, the question we most commonly get these days, as Dana sort of touched upon, is, do you have capacity and when is it going to be ready? And then who are the people that are going to execute on my program? And what is your culture for person and plant and partner and plant? So those are the questions we most often get. What's interesting about what we're bringing to the table and what we talk about often with clients is that we can accommodate everything from plasmid DNA, like I said before, to viral vector production, to cell processing, all in different buildings, per Dana's point. But, we do have a 2.4 million square foot campus, as you pointed at the top of the call. And what's super interesting about cell and gene therapy is that 88% of viral vector manufacturers outsource and 52% of cell therapy manufacturers outsource, at least at some point and in some way. Half of those numbers approximately use a hybrid strategy. So what's interesting about Center for Breakthrough Medicines is that we have a partnership with Discovery Labs, which is a real estate company where we're located, and we really have unlimited expansion capacity, up to 10 million square feet, all in King of Prussia, Pennsylvania, which the people on the call know, at 20 minutes outside of Philadelphia, really in the heart of what we're calling Silicon Valley. And it's really nice to be able to bring this offering to the table. It's very unique from other CDMOs to have a property company component. And what's enticing about it is that a company is able to scale all the way from idea to commercialization on one location. So that's and you can cohabitate with your CDMO. So we're addressing some of the concerns that you brought up, Ross, but also bringing this really unique aspect to the table for companies that want to outsource, insource, or co-locate and grow in place.
While we're on that, so we want to kind of bring this around, but I think you hit on something I want to get input. So I'm going to ask Audrey and Dana and Graham, think about this, and then we'll go. So considering the commercial successes of Cell and Gene and the lessons learned and all this other, and the great pipeline and all the plans ahead, in a few, what in your mind are the challenges in timely commercialization? So Audrey, you can lead off, and then Dana, and then Graham. Okay, how's that?
Yeah, that sounds good. Developing commercial grade processes, analytics, materials, and platform as early as you can in the process of the path to commercialization is key for avoiding problems down the road. I think analytics have been the hot topic recently with potency assay, but certainly across the board, having that GMP grade materials, know-how, processes, methods, and analytics are going to be key to getting commercialized.
Dana?
Yeah, so my experience was, transferring technology out of GMP in an academic facility to a major pharmaceutical company. And so, and what I witnessed as a key challenge there is that, this isn't about just transferring over some SOPs and having a couple of meetings around it and like, good luck, guys. And it was really, really intense in terms of bringing the teams together from both the academic facility as well as the commercial facility to figure out like, how does that actually transfer over into a scalable commercial pipeline? And it just wasn't a simple, let's hand over some SOPs. And I think what the field would really benefit from is when a step like that needs to happen, it's really coming together of two worlds of experts where you're transferring knowledge from one to the other and training each other and working together over a bigger chunk of time. And I think that's going to, I think that's going to help things become more seamless overall as we move forward.
Graham?
Yes, I agree with both of what Audrey and Dana had said. Maybe just add a couple of things. One is there's a dynamic tension between clinical and CMC development. The patient is waiting. There's a high pressure to deliver a product, but then there's a CMC requirement. And then so there's a need to coordinate between those two groups to assure the most timely commercialization possible. And then there's just a couple of tactical things that I brought up earlier that I'd just like to reinforce. Access to manufacturing capacity, planning that early, making sure that there's a place to manufacture when that's needed. And then what's become quite problematic over the last year or 2 years is availability of raw materials and consumables, and planning early for that as well, because you don't want the lack of a single-use disposable bag to hamper your ability to commercialize. So there's a lot of planning necessary to do this well and to address timeline issues.
Thanks, Graham. And I think I want to thank everybody on the team here. Again, I think we could have gone on for another 2 or 3 hours, but I think we can call it quits at this stage of the game. I want to thank everybody again. I also want to mention to our folks that are listening that they will hope to see everybody on the floor at Interphex, October 19th through the 21st at the Javits Center in New York City. And with that, I want to wish everybody take care and talk to you again next year. We still have to talk about. Take care, folks.
Thank you.
Take care. Thank
Bye-bye.
you.